All right, what's going on, folks? I hope you all can hear me, and it's not too loud or too soft. You can let me know what's up. I think I can see that it's working on my end of things, so hopefully it's working on your end of things as well. But so yeah, here we are for Unit 1, AP Biology. If you're taking this class, you can use this as a review. If you're one of my students, I hope that you will use this as a review. And my goal is to just let you ask questions. It is working. Awesome. Thank you. So yeah, if you guys can just ask questions that you have, and we'll spend this time talking about stuff. And if y'all don't have any more things, then we can call tonight. And I'll go play some Fortnite. So does anyone have any questions for me? And I'll go I just went and looked at my stream, see if it was like looking clear. It does look clear. Everything looks nice. I've hardly been on this end of streams. Normally I am doing the streaming. So what is the charge of the N terminal and C terminal? That's a perfect question. Let's go look at that. All right. Okay, so good question. So here's a amino acid. And so amino terminus is the same as N terminus, right? And so we could just write that and that'd be the same. And carboxyl terminus is the same as C terminus. All right, and so what will happen, so this is your standard amino acid. This hydrogen here will let go. It will it ionizes really easily. And so if a positive goes away, it's going to leave this negative. And so the C terminus is going to be negative, and the N terminus is the opposite. It's going to go out and get additional hydrogens. And so it's going to make that, it's going to tend to be negative. And so that is why the C terminus of one amino acid is attracted to the N terminus of another amino acid. And you can see that really well. I love this picture, by the way. If you ever have to memorize the amino acids, these are great pictures. I got them because they're probably in the textbook that I learned from. And so you can see that here. There's the carboxylic acid or the carboxyl terminus, and then you see it's negative, and it wants that positive, right? And rather than forming an ionic bond or a hydrogen bond, remember water will get pulled out of this system, and it will cause dehydration synthesis. They'll join together, and that's what you see right here, right? Water's being pulled out. So you have two amino acids joined together, and there you have water. Yeah, this is the same question that that both of you guys had. Basically the same idea. Don't think of it. Don't try to get in the weeds with it. It's mostly just, if we go up here to nucleic acids, right? They have some directionality too. We talked about the 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. This is just that version of that. It's just its own special version of that, And the same thing with carbs. They're going to have their own thing, except for it's a little simpler because there's not as many pieces. It's just CHO. Lipids, not as much. They're pretty simple. Though phospholipids, There was a couple questions in the practice problems about the hydrophilic portions or the nonpolar portions not wanting to be near water, and the polar portions wanting to be near water, and that's why they'll form this bilayer. So it's all the same idea. It's all the same idea leading to that shape or whatever. Great questions. Hopefully that answered it. Let me know if it didn't. Wow, that tea is really hot. I took out a little bit too much of a swallow. One of these days I'm just going to spit tea out on the stream. Not going to be today. Yeah, and another thing I encourage you to do, if you're kind of struggling with some of that, yeah, this is a lemon and ginger tea. It's what the body needs. But if you're struggling with some of this, some of these concepts, just go look at other pictures of it. Like just find the weird pictures. Like here, I just found a picture of some amino acids. I'll post it in here and we'll see. Yeah, because I think it's good to just see things a different way sometimes. Yeah, I'll do that for sure. Teach each macromolecule and its elements. Yep, I will do that. All right, yeah, and I'll talk about that one too. Good. All right, so let's do the contents. We're gonna start with carbs. What's in a carb? Carbs are carbon, hydrogen, and oxygen. That's it. And the rule that I have always used when I think about carbs is C equals O. Those are my mouse drawings, so you just got the deal. Are there any other noteworthy directions besides? No. C terminus, N terminus would be 1, 2, I guess, for the amino acids. So that's carbohydrates. C is equal to O. So let's test it out. There's 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And what's the chemical formula for glucose? C6H12O6, right? Even like sucrose is C12H22O12. And so, even though the H is not double, the O and the C are still the same. And so carbs, C and O equal. Lipids, CHO also, but notice a whole lot less oxygen. Right? A whole lot less oxygen, and mostly these long chains. These are called hydrocarbons. I didn't really use that word. Thanks. Draw and hold to enhance your shapes. Maybe I will. Let's draw and hold and enhance my shape. Nifty. All right, so this is a hydrocarbon. Why is it called a hydrocarbon? Because it's hydrogen and carbon. And so lipids will typically have these long hydrocarbons. Here you go. Not as long, right? Triglycerides. Something similar to that. So that's lipids. Also CHO, but a lot less O. Notice the C is not equal to the O here. C is not equal to O. All right, let's see if I can do that. It doesn't use the nifty shapes for that. Well, it did straighten that line for me. All right, so now let's do proteins. What do you notice about proteins? There's one thing that sticks out. It is nitrogen. Nitrogen is present. And I'm going to say something in nucleic acids just a moment that's going to make, oh, you're like, well, that's nitrogen too. But there's one thing in nucleic acids that's always there. And amino acids are always going to have this R group. Sometimes there'll be an R, like this box with an R. And sometimes it'll be some kind of random thing, right? But this amino acid's always going to look like this. This portion of the amino acid is always the same every time. And this is the R group is going to be a variable, and that is what's going to like change that up. Good. And then nucleic acids is next. Nucleic acids always have this particular shape to them. So let's go here. They all, look, there it is. Look, wait, no. There it is, look. Well, there you go. That's a short one. Look, there it is. Look, there it is. They always look the same. You have this PO4, this phosphate, 5 carbon sugar, 1, 2, 3, 4, 5, and this nitrogenous base. Sometimes the nitrogenous base will have 1 ring, sometimes it'll have 2, depending on which one it is. We didn't really talk about that because it's not important for this particular unit. And so, yeah, nitrogenous bases always have nitrogen, or nucleotides always have nitrogen, and they also have phosphorus. That is the thing that they're always going to have because of that phosphate. No other micromolecule has phosphate. So that is that question. The con question, which the following biotic best demonstrates water's adhesive. Yeah, so capillary action. So think of a tube. All right, so think if you were climbing a rope, right? If you were climbing a rope, what would you need to do? If you're good at climbing a rope, you understand this. I'm not good at climbing a rope, so I don't understand this. But if you're good at climbing a rope, you have to reach up your arm, right, and grab a hold of the rope, and pull it down. That's the adhesive part. And then you also have to pull yourself up, right? And so that is the cohesive part. That is the water is cohesive, so it pulls itself up, and it's adhesive in that it grabs a hold of the tube and pulls itself up the tube. And so that's probably where they're getting that from, I'd guess. I didn't see a lot of the con questions this time. There wasn't a lot of people doing them. A lot of people were doing AP Classroom, which is fine. But normally I see more of those. Adhesion sticks to itself. Yeah, sticks to self. Adhesion does stick to self, but that word is specifically used for think like adhesive tape. What does adhesive tape stick to? Anything, right? And so, and we talked about the noteworthy thing, the directionality. All of them are going to have some directionality, but 5 to 3 is like the thing that really is kind of different and new with this, and then less so the N-terminus, C-terminus. And this particular picture, I didn't really spend a lot of time on. But what I want you to see with this picture, this is the N-terminus, and this is the C-terminus of this amino acid, right? This is the C-terminus, and this is the N-terminus of this other amino acid. And keeps, there's a couple, there's actually 3 amino acids here, right? And each one has a C and an N-terminus. And so I think that's important. How does water's polarity, hydrogen bonding contribute to emergent properties? Yeah, I can give you an example of that. Okay. So here's water. All right, so I've had several people ask about this idea of emergent properties. I think it's more of it's kind of a background concept, which is fine. And so let's go back up here. Here is a neutral, or a neutral atom, right? So 2 neutrons, or 2 protons, 2 electrons, it's neutral, right? And so this oxygen is also a neutral atom, right? 8 protons, 8 neutrons, 8 electrons. And these hydrogens are also neutral. However, when you combine them together, they will share. So they have properties as a neutral atom, right? They have properties. But when you combine them together, they'll share all 10 of those electrons, 8 here, 1 each here. And because oxygen is so much bigger, there's this new emergent property that happens, and it's the unequal sharing of electrons. They're still sharing them, but they're sharing them unequally, and that is where polarity comes in. It emerges out of this thing. It's it's like the puzzles that we were putting together. You get the little picture of the little girl in a window, and you're like, oh, this is going to be happy. And then you put it together, and you realize that she's in the burning building. That was a property that emerged that wasn't there before. And so hydrogen and oxygen, neutral atoms, add them together, make water. Still neutral, but there's this emergent property called polarity that happens that wasn't there before. And so that's what we mean by that. Primary, secondary, tertiary. Yeah, we can look at that. Faux show. Let's go look at that. I think it's this one. It is. All right. Primary structure is just amino acids. Here you go. This is a primary structure. I've made a mess of this. But this is a primary structure as well, right? It's just 3 amino acids. Just a chain of amino acids, and each amino acid is connected with a covalent bond. And how the covalent bond made? Dehydration, synthesis, right? Pull water out, add 2 things together. Do that a bunch of times. You get this Mardi Gras beads right here, right? And because of these pluses and the minuses that are going on, because of these oxygens, because of these nitrogens, because remember up here, what happened when you got a bunch of oxygens, nitrogens, and hydrogens together? Well, hydrogen bonds form, right? So because those hydrogen bonds form, the same things are going to happen here with these amino acids. And they're going to kind of curl up, or they're going to curl up like a helix, or they're going to like fold up like a sheet, like the pleated sheet is what this is called, which is just a fancy word for a fold. And that was the thing I showed you with the pipe cleaner, right? Where I like had the pipe cleaner and I twirled it around my finger and like showed you how it made this complex kind of structure, right? And so this one, whereas these amino acids are connected with covalent bonds, the bonds that are like holding this spiral together, like right here, and these folds together, those are hydrogen bonds. So secondary structure, hydrogen bonds. But notice there's some 3-dimensionality that's starting to happen. Well, when those secondary structures start to fold up on each other, then you lose your whole picture. No, wow, where? I got too close to the sun. All right, and so these pleats and these helixes will start to like bind together, right? And this is called a tertiary structure, or third level, and the tertiary structure will actually form covalent bonds, and these different, and one of those bonds is called a disulfide bridge. That's one that you'll see a lot. So some proteins are done, like it's a done protein as a tertiary structure is done. Others, multiple tertiary structures will join together to form a quaternary structure, and they'll join together with different kinds of interactions. We don't need to know those. The ones that I mainly were concerned with was covalent here, hydrogen here, and this disulfide bridge here. So, and this is when I talk about proteins having a shape, their shape determine their function. This is the process of it going from a string of beads to something that has a shape that can do a job. Explain how structure relates to function with proteins. So that was that with proteins. So lipids, I'll use a great example for lipids is phospholipids, of course. So here's a phospholipid. Phospholipids are part of them is polar and the other part is non-polar. So up here you have this polar portion or hydrophilic portion, which means water-loving. And down here you have these fatty acid chains, just long hydrocarbons, right? And they're non-polar. And so how does that determine its function? Well, there was a question in AP Classroom that gave you a ton of information, and then you didn't need any of it. You just needed to know what was polar and nonpolar and what happens when you put it in water. And so if you put a bunch of these things in water, what's going to happen? Well, the hydrophobic portions are going to get away from the water, right? And the hydrophilic portions are going to want to get to the water. Well, and so they form this sort of structure. And this is called a phospholipid bilayer. Whereas you have water out here, right? That's H2O. And we're going to pretend that I wrote H2O down here at the bottom as well. And see, this is water. There's just a drop. There we go. There's water down there also. And the polar portions of the phospholipid love the water, right? But the hydrophobic or the nonpolar portions hate the water, and so they'll turn in toward each other. And this is a nice barrier. Nothing can get in here, and nothing can get out unless there's some sort of like special passageway or something, which we'll talk about more in Unit 2, I think. So yeah, and so that was lipids. Carbs. Remember we talked about the wow, that's square. Sorry, I got distracted with this square. We talked about these carbs, yeah. So the shape of how starch is all the, just like holding hands, right? So I've always thought of that, and they're all pointed down. And starch is this molecule over here, and glycogen is very similar, and it's branched. And the purpose of this is to store glucose. Plants will store glucose using starch. Animals will store glucose using glycogen. Cellulose does not form that structure. It forms these nice, neat, almost like bricks, right? Because cellulose is used to build things like tree bark, and it's what make like blades of grass or whatever stand tall. Well, look at cellulose. It's opposite, right? And so you can almost see it kind of stacking in there, like a, you know, if you were to have another string of cellulose, like right here, you could like picture it just stacking in rows. Right, and so whereas starch, you eat starch like a potato or a piece of bread or something, body knows exactly what to do with it. There's an enzyme for that and everything. You eat a piece of cellulose, like if you just went and like just chomp some tree bark or something, that bark's not going to do much inside your body because the body doesn't know what to do with this shape, but it does know what to do with this shape. So that's an example. There's lots of examples of that, but that's just one. And then lipids, I mentioned phospholipids, proteins, nucleic acids. Nucleic acids are kind of their own thing. We're going to spend a whole unit talking about DNA. But one interesting thing about its shape, and we didn't really talk about this, and it's not that important, is that, at least in this unit, but the phosphates are really negative. And what do negative charges want to do? Well, they want to get away from each other, right? But they can't because they're connected right here. This sugar phosphate backbone. And so, what's the only way they can get away from each other? Is they'll like twist. And so that is why DNA has this helical structure, is because the negative nature of DNA. DNA is extremely negative. We will actually do a lab this year in the spring semester, where we will use DNA's negative charge to do a lab, which is pretty cool. Question, what macromolecules are polar or non-polar? So yeah, it's going to depend on the molecule. Lipids are generally non-polar. Carbs, I just hate to make distinctions like that. Carbs are generally polar, but not. So like, okay, here's an example. Glucose is polar. You put some glucose in water, it'll dissolve immediately. How do you know? Well, look, this big oxygen's like, I want electrons. And these hydrogens are like, I don't. and so it will dissolve. Now it's not going to like come apart like salt. It's not going to ionize. But, and I'm saying too much, but glucose is generally polar, and so it's going to dissolve in water. Nucleic acids tend to be polar, more negative. Proteins are all over the place. There's no way to really talk about them that way because they're so all over the place. But polarity of water, that's a big idea, and so I would stick there. And usually any other place you're going to be like given the info. What did you not cover that's going to be on the test? So yeah, that's a good question. I'm pretty sure I covered everything that's going to be on the test. No secrets. Now, there's probably some things that, you haven't seen before, but you can use the things that you know in order to answer questions about the things that you don't know. And that is one of the great things about this class, is that you get to learn that skill. It is a good thing. Going here and cleaning all this up for myself. So yeah, nothing that I can think of that it's not going, unless y'all watch the videos and found something that exists that I don't know about. Perhaps there's that. But I can't think of anything. I looked at the test today, and it's good. I'm not worried about it. So you'll be fine. Just don't overthink things. The same advice that I've always given you. So yeah, that's a good question. I think that things that students struggle on the most is, as soon as I said that, I wanted to talk about my honors biology test today, and the fact that, this is a good example of how sometimes it's harder to unteach something. So in my honors class, in their notes, and you guys can attest to this because it's in your notes from honors too. Some of you still have your notes. You can go back and look at 1 point, probably 1.4, and even maybe 1.1, but most definitely 1.4. And you can look where it says crystalline structure. I'll talk about crystalline structure. Where it says the dependent variable is what's measured and the independent variable is what is manipulated. And I said those words and it's there, and yet, no kidding, 80% of my freshmen told me that the independent variable can't be changed and the dependent variable depends on the independent variable. And so it's just an example of something. And so I think what do you struggle with is something you can ask yourself. It's kind of a reflection. What are things that you thought you knew before we started this unit, and then you heard something different that changed that, and then are you going to then rely on those things that you thought you knew? And ask yourself that question. Nothing comes out to me on this particular unit of something that students really struggle with. It's a lot, and so I think that's the big thing. Crystalline structure typically has to do with like ionic bonds. So let me see if I can go out here and find one. It's not something we covered. All right. So basically it's the idea that, so here's sodium chloride, like table salt, right? Sodium ionizes, gets rid of an electron, chlorine ionizes, picks up an electron, and so they like each other. And when there's no water, they will form this crystal structure, which we're familiar with, like salt crystals, right? But if you have water, and this is actually helpful for our discussion, if you have water, you have O here, and you've got two H's, right? Well, this oxygen is negative, and the H's are positive. And so what's going to happen to this salt crystal? Well, the positives are going to go and surround this negative, and the negatives are going to go and surround this positive, and it's going to pull the salt apart and or dissolve it. And that's like the we talked about water solvency. And then what happens if you take the water out? Well, then those salt crystals will form back. That's what's going to happen. Talk about carbon's importance. Yeah, let's talk about carbon's importance. Let's pause for a moment, see this in all of its glory. I mean, that's better. Okay. There we go. Okay, perfect picture. So if you just look at this picture, and I were to ask you, why is carbon important here? You would say, well, it looks like it's forming like some of the... That's not what I expected. I thought that was a highlighter. What is that? Here's the highlighter. Looks like it's forming like some of these, like almost like what I would think of like a backbone or like a foundational kind of structural component of these molecules. And so that is what I would say, that carbon forms a structural component. And why is that? Well, look at carbon. It can form 1, 2, 3, 4 bonds with other things, and those are covalent bonds, and so they're really strong. And sometimes it forms bonds like this is still 4 over here, right? 1, 2, 3, 4. It's just double bonded, and so it forms that. It's really strong. Building block, perfect. Yep, it is the building blocks. The other ones are, and the other atoms are building blocks too, but the carbon is always going to be the thing that you find in the center. It's like it's like the floor joists of the house, or like the roof things, or the studs in the wall. It's the skeletal component of molecules, and you can see that most of the time. So, like, perfect example here is this lipid. They don't even bother putting that on there because it's just known. This carb, see, this black is all carbon. It just weaves itself throughout. Even with amino acids, carbon is that component. Look, nitrogenous base, carbon. It's just holding everything together. How is it unique to form? That's why, because it's so, it's just so universally able to bond with things that it's able to do that. It fits with everything. It's like the Lego block that. So imagine thinking, man, if I just had this block and that carbon always fit there, or if I just had this one, oh wait, here's carbon and it also works. And so it just kind of works every time. There's not a situation. Well, I mean, there are situations where it wouldn't work, but. So yeah, if you take organic chemistry in college, some of you will. And organic chemistry is literally the chemistry of carbon. And it gets complicated. It gets pretty complicated. I did not do very well in organic. I had to save some for the next students. That is for sure. All right. You guys have any other questions? Any other thoughts? Any other concerns? Any other things that are just eating away at you. All right, let's see. Yeah, let's see. Some of these have questions on them. I used to use them in class, but I stopped using them because some of them are just bad questions. And so, let's see. Perfect. Okay, here's one. So this one's not. So, okay, so application. Perfect example. You're going to be usually given a sort of picture or something, right? And you got to like figure out what's going on. Water, there's some molecules, right? And then there's some other molecules. Which of the following best describes the properties of the molecules indicated in Figure 1? See, this question's just bad. Oh, it's going to separate them. Molecule B has hydrophilic properties. And molecules A and C have hydrophobic. That's wrong, right? Molecule B has both hydrophobic and hydrophilic properties. I like that because B has there's hydrophilic and hydrophobic, right? This is phospholipid membrane. And both A and C are only hydrophilic. Yeah, C is not though. B and C have both hydrophobic and hydrophobic. I think D is the correct answer. Look up. So yeah, see, I had to kind of work through that, and you're basically eliminating parts that are right and wrong, and kind of that's what I mean. Or they may give you something that's completely weird. Like there was one question in I think it was AP Classroom that had I helped a couple of you guys with it, and it was like a long train of amino acids, and then there was a mutation on position 502 or something dumb, and there was another, and it showed that one. It was different, and then it like, what would happen? And it was like it wants you to like get all up in the story of the mutation and the little pictures, but it literally is a question of do you understand hydrophobic, hydrophilic, and what would happen if you put it in water? And that's is a question of that, and that was all it was. And so that is the skill that you really will develop over the course of this year, is like figuring out what is this question asking, and then being able to kind of like weed through the garbage to get to that. In DNA, are new ones added to the 3'? Yep. And form bonds to the 3. Yes. And covalent bonds, correct. Yeah, covalent bonds. And remember, water pulls out water every time. But I have a picture of that. Wait, no, it would be here, I think. Yeah. This is actually it right here. This shows you that too, right? Because that's wait. Okay. So 5'3", it's being added to the 5 or to the 3' end, right? Right there, perfect. Pulls H2O, or in this case they're being split. So let's go the opposite direction. All right, so it's added to the 3' end. It's going to pull water out, and it's going to combine them to make a single one. How do they connect to what we talked about at one point? Nucleic acids. What did we talk about nucleic acids at 1.1? I don't know. I mean, they're just... How do they connect to water, I guess, is like, water's used to break them and form them, I guess, would be the connection. I think the connection, like, it's just like, chemistry of life. It's kind of trying to throw a bunch of stuff in there that's kind of, fits, I guess. Oh, look, that line straightens out. I love that. How are all three molecules hydro that little heart's in my way. Hydrophobic, okay. I don't think, let me go back to that question. Okay. So B and C have hydrophobic properties, right? Because they go into the middle where this hydrophobic portion is. But all three are hydrophilic because all three touch water in some way. I mean, the concepts connect. The only way the concepts connect is like just understanding. I don't know. I guess water connects all of it. But other than that, no, not really. Water is kind of water deals with all those macromolecules, and so that is the connecting factor. The fact that hydrolysis, dehydration, synthesis works for all four classes of macromolecules. That's like what Ava said. That's how I would make the connection too. Definitely a big idea. Yeah, it depends. I mean, or polarity or lack thereof. Yeah, and some molecules are so big that there's like nonpolar and polar portions of those molecules, kind of like what you see here with these. And so, like, this molecule B is representing some protein, I'm guessing, that's going through the hole. And so it's both of them. It's polar and nonpolar. Well, actually, in the next unit, I'll give you a word that means polar and nonpolar, but you don't need to know that word now. The test does have an FRQ, and it is not a graph. It is just a written. Though I do think the FRQ is going to have you like actually write on the test, because you have to practice that, because one of the questions on the exam will have you write on the test, and that will make sense. What I mean by that is like write on. There'll be a drawing, and it's going to ask you to add something to that drawing, which is always one. There's always one question on the exam every year that asks you to do that. So there's going to be some people that are going to come in later and watch this. So what questions do you think they might? What questions do you anticipate them asking? So we could help them out, maybe. Who that is? Who that is? Yes, tomorrow. It's nothing, though. It's literally nothing. It's literally nothing. It's nothing. It's literally nothing. I don't know how to tell you. It's a good thing. Many people have taken this test and survived. The difference between dehydration, synthesis, and hydrolysis. That's a great question. Let's talk about it. I need a picture that has kind of both. The mono and poly of everything. Yeah, the poly is the name of them, but I would definitely know the monomers. Like monosaccharide, nucleotide, amino acid, and fatty acid. I would definitely know those. And we'll talk about the difference between those bonds, and that's good. Those are good questions. So let's I'm going to see if I can find a question that has both of them, actually. Or a picture, sorry. Yeah, here we go. Perfect. Okay. Because I think that just seeing them together will help. Okay. All right, so this is great because this is just two carbs, two little glucoses. And you have two glucose, two monomers, right? Two monosaccharides. Here's one. That's a big yellow pen. Pineapple pen. All right, so 1 and 2. And now they pull water out and then they combine together. So this is one molecule here now, right? And here is water. Touch on entropy. Yeah, I can do that. And down here, it's the opposite. You take this thing here and you're adding water to it and it separates them, right? And so that is the difference. But they are linked, right? They are the opposite of each other. And I think that may be an important distinction to help you all. They are the opposite reactions. One is take water out to make things. One is add water to break things. And a covalent bond, good question. So these are covalent bonds here, right? Because these are carbons right here. Carbon. I don't know why I chose to make that C backwards, but that's what happened. These carbons are sharing electrons with this oxygen. And so it's a physical connection, just like the whiteboard in my room is sharing a screw with the wall, right? There's a physical connection. Versus a hydrogen bond, which is not on this one. Is perfect. See these two water molecules? They're not actually sharing anything with each other, but there's a slight charge on this oxygen and a slight positive charge on this hydrogen, and they're attracted to each other, just like the erasers on my whiteboard. The whiteboard and the erasers are not exchanging things, but they are attracted to one another. And so that is the difference. We call that a bond. I don't call it anything. I'd have called it something else probably if I was given the choice. But we call it hydrogen bond because that's what it's called. If this was a covalent bond, then you'd expect to see a physical connection here, right? A more physical connection. Yeah, we didn't really talk about directionality in lipids because it's not as much of a thing. So I'm not that worried as much about that. Let's see if I can find. For lipids, it's just you're just not going to see it as much. Understanding the difference between the saturated and unsaturated would be great. I think would be good for that. Does hydrogen have to be present for a hydrogen bond? Good question. So typically it is because it represents that because hydrogen is so small, right? And there's other molecules that are going to pull its electron away. And essentially what's happening is I don't really have, let me see if I can find a good picture of a hydrogen bond. Because like a 3D picture. Yeah. I always have to look at these pictures. That was okay. So, okay, here we go. No picture, but I just don't remember how I was going to talk about it. So right here, imagine a situation where there's equal sharing, right? And anywhere, if there was equal sharing, you could touch this water molecule anywhere and it would have the same sort of charge, be neutral. Well, it's not. And so the electrons are going to be up here most of the time. And so what does that then expose down here? Well, if hydrogen loses its electron, what is left? Just a proton, right? And so what charge does a proton have? It's positive. And so that is the only thing that's left. And that's kind of a way of thinking about it. And so that's a Krug question. Because but I'm assuming, yes, I'm assuming there has to be hydrogen just because of the size of hydrogen and what bigger molecules like nitrogen and oxygen are going to do to it. It's going to rob it of its electrons. But that's a Mr. Krug question. But I'm assuming that he would say yes on that. For sure. Yeah. Anything else you guys have before we close it up for the evening? Hopefully this has been helpful for y'all. I enjoy doing this quite a bit, so yeah, that's, I assume, because of that. Oh, entropy. Thank you. All right, so entropy is just this idea. Gives me an opportunity to come back here. Okay, so actually we'll go here first. Wait, that went, I didn't mean for it to do that. All right, here we go. And we get to wonder what is on this thing's shoulder again. Okay, so in a perfect world, this gazelle eats that grass, and it eats that grass, and it gets 100% of the energy out of the grass, right? In a perfect world, but it doesn't. Now, you guys remember this because of, you remember in ecology, the rule of 10%, right? The gazelle is going to use 90% and it's going to pass 10% on to the next generation, right? Remember that. Part of that 90 is entropy. The gazelle's interactions are not 100% efficient. All right, I'll give you another example. It's like a car engine. Well, no, we'll just stick with a gazelle. If you went up and felt of this gazelle, if it let you, and it might because it needs some shoulder surgery here, it would be warm, right? What is that? That warmth represents inefficiency in the interactions, in the reactions. And so it releases heat, and so it's warm. That heat is a sign that metabolism is currently happening inside that animal's body, right? And it eats the grass, and it gets some of the energy that the grass had, which the grass is able to get some of the energy that the sun had. And these cheetahs then are able to, after making a pact with their Dark Lord masters, able to get some of the energy out of this gazelle, right? But not all. And so if you keep saying some, what are you going to have left? Well, there's nothing. Eventually you're going to have nothing, right? And so that is entropy in a nutshell. We will talk more about that as we go along throughout the year. It's a big idea, for sure. If the cheetah's eyes are red, what color is the gazelle's eyes? Rainbow? Why is there a rainbow? That was too big, too deep, and too greedily. Oh my. Okay. That looks rough. And I still wonder what's going on here. Maybe it's another gazelle back. Maybe it's, I don't know what's going on that shoulder. All right. Any other questions, thoughts? There's a definite French bee right there before we close up for the evening. Ocean, man. This is an ocean. I can tell I'm starting to get I'm starting to get tired a little bit. Well, I'm glad this was helpful. I'm glad it was. I'm glad that because I like doing it, so it's fun for me as well. And I'll plan on doing it for the other units that we have. I'll be at school. It's at 7:30-ish in the morning. So if you have questions, please come by. Exclusive. No, there's really nothing. There's no secrets. It's all there. No exclusive. The test is going to be. There won't be any surprises. You'll be like, oh, okay, it's not going to be bad. But yeah, I'll be at Air in the morning, and so come see me. 7:30-ish. We'll get started as soon as the bell rings, and it won't be bad. And then it'll be over, and then you'll have a nice 3-day weekend to enjoy. So I'm gonna go ahead and be done. Thank y'all for stopping by, and appreciate y'all supporting my channel, and glad that I can help y'all out as well.